Hello and welcome to this revision video for Biology Paper 1. In this video I'm going to go through the extra content that is needed for separate scientists only. So if you're one of the students that takes the 6 1 hour and 45 minute exams, that means you'll need to know a little bit of this extra separate science content. Remembering them for Paper 1, there's 4 topics: cell biology, organisation, infection and response and bioenergetics. I'll just go through the extra content in each of these topics. First of all then, for cell biology, the separate only contents are about bacteria, how they divide through binary fission, preparing an uncontaminated culture of bacteria, and then using that bacterial culture to investigate the effectiveness of different antibiotics. First of all then, we'll look at how bacteria divide through binary fission. So if we start here with one bacterial cell, if you remember, they're prokaryotic, so they'll have a loop of loose DNA, and they might also have 1 or 2 plasmids as well. If the temperature is ideal and there's enough nutrients, then these bacteria will divide. So what they'll do is they'll first of all duplicate their genetic information, so they'll make a copy of the circular DNA, and they'll make copies of the plasmids as well. Then the cell will elongate and divide into 2, and when it does this, because the loose DNA goes to either side of the cell, each new bacterium will each have a circular loop of DNA, but the plasmids won't necessarily divide equally between the 2 new cells. So in this one, for example, you could well have 3 plasmids and this one just 1. Bacterial cell division by binary fission is very quick. The time for each cell division will depend on the bacterium, but it can take around 20 minutes for each cell division, so they can multiply really rapidly. So after another 20 minutes you'd get another round of binary fission, the loose DNA and the plasmids duplicating, and then the cells dividing. So you might have to do a little bit of maths as part of this topic. This question here says a bacterium divides every 20 minutes. Calculate the number of bacteria present after 3 hours. So each cell division will take 20 minutes, at which point the number of bacteria will be doubled. So first of all, you need to work out how many cell divisions there will be in 3 hours. So if we convert this to minutes, there's 60 minutes in an hour. So 3 hours is 180 minutes. And if we do 180 divided by 20, because this is our cell division, we can work out that there will be 9 cell divisions in that time. So a really quick way to then calculate the number of bacteria is to do 2, and it will always be 2 to the power of, in this case, 9. So it will always be 2 to the power of the number of divisions that you will have within that time frame. So in this case, 2 to the power of 9 will give the answer of 512 bacterium. So if we started with 1 bacterium and it divided every 20 minutes, the number of bacteria after just 3 hours would be 512. If you can't remember to do it this way, you can do it another way by just showing each time that you would double the number of bacteria. So if you started from 1, after 20 minutes you'd have 2, another 20 minutes you'd have 4, another 20 minutes you'd have 8, and so on until you got to your answer of 512 as above. So if you can't remember to do 2 to the power of the number of divisions, then you can do the slower route of working it out to get to the same answer. If you're doing higher tier, you may also be asked to express your answer in standard form, so look out for that. They might not make it obvious. Underline it in the question. So in this case, if we're looking to put this into standard form, it would be 5.12 times 10 to the 2. Next, I'll talk you through preparing an uncontaminated culture of bacteria. This is where you want to seed an agar plate with an even spread of bacteria and make sure that only the bacteria that you want to grow on the agar plate will be growing there. So you start off with an agar plate. This is a Petri dish with agar jelly all the way in it. So that provides the nutrients. And first of all, you need to sterilise the agar jelly and Petri dish to make sure that there are no bacteria on there to start off with. You then need to have a little metal loop which is going to transfer your bacteria. This is called an inoculating loop. And you need to sterilise this by passing it through a Bunsen flame to kill any existing bacteria that are on the inoculating loop. Then, when this is cooled down, dip this into your bacterial culture that you want. So here we've got a small test tube with our bacteria culture in there. And then once we've dipped it in, we will spread it evenly across the agar jelly and allow it to grow. Final couple of things you want to do. You want to put some tape on the lid once you've put the lid back onto the Petri dish to stop any microbes from the air going in and growing on that plate as well. So we've got tape here to stop the microbes from the air entering, because we want it to be uncontaminated. We only want the bacteria that we've put from our culture to grow on here and nothing else. So we put tape on it. We also store it upside down because we don't want the condensation to drip back onto the agar jelly. So if we leave this in a warm environment, there'll be condensation on the lid, and if we left it this way up, that would drip down and ruin the bacterial culture. So here we have storing it upside down to stop the condensation from dropping onto the agar and the bacteria. The final thing that we need to remember is that we're only going to keep this at 25 degrees C. So in the lab and in the school, we can only keep it at this temperature, because at this temperature we can prevent more harmful microbes from growing. If we get higher temperatures in industry, they can do it at higher temperatures, but they run the risk of harmful microbes growing on that agar plate. So now we've grown our uncontaminated culture of bacteria, we might want to then investigate the effect of different antibiotics on the growth of the bacteria. And this is a required practical for separate-only scientists. So here we have our agar plate. It would be seeded using the method before with an even spread of bacteria all across the plate. And then on top of the plate, you could put paper discs which are soaked in an antibiotic. So this could be one antibiotic, we'll call it P, and this could be a different antibiotic, let's call it S. Now antibiotics are very specific to treating a particular bacterium. So we might want to investigate which antibiotic is best at treating the bacterium that we've put on our agar plate. So here we put down our paper discs which are soaked in the antibiotic, and we may also want to put down a control disc as well, just to prove that it's not the paper disc that's preventing any bacteria growing. So we might just want one that is soaked in water, whereas the other ones are soaked in an antibiotic. And now we've set this up, we would leave this at 25 degrees C for maybe 24 to 48 hours. And what we would see on our agar plate is we would see bacteria growing all over the agar plate, okay, evenly where we spread it. But around the antibiotics we will see areas where there are no bacteria growing. On one of them, that area will be bigger than the other one. So we can see here, we call these the inhibition zones or zone of inhibition. So we can see here, this is an inhibition zone because that's where bacteria have been inhibited. They cannot grow. And equally, there's one just here. So this would show that the effectiveness of the different antibiotics, this one was P and this one was S, and we can see here this antibiotic is much better at preventing bacterial growth here and killing the bacteria than this one around here. But we might be asked to use data to prove this. So what you'd have to do is you'd have to measure the area of the inhibition zones using pi r squared. So what you need to do with your ruler is measure the diameter of your inhibition zone, but don't forget you need to then divide that by 2. So let's say, for example, if we measured this and that was 10 millimeters, our diameter. To work out the area of the inhibition zone, we'd have to calculate the radius, which is half the diameter, so the radius would be 5 millimeters, and then we do our pi r squared. In our calculator you can use the pi button, or if not, use pi as 3.14. So if we do pi multiplied by 5 squared, we will then get our area of 78.5 millimeters squared. And then we would do the same for the other inhibition zone as well, measure the diameter of that, halve it to get the radius, and do pi r squared to prove which antibiotic is best. As well as using pi r squared to measure the area of the zone of inhibition, they may well ask you to estimate the area of some bacterial colonies. So if you see lots of colonies like this, it's quite difficult to calculate the area of all of them individually. So you can calculate the area by measuring across with your ruler the diameter and then dividing it by 2 to get the radius and doing pi r squared. So they may ask you to estimate the area of a bacterial colony, and if they do that, use pi r squared. For topic 2, which is organisation, there are actually no separate only topics for this section, so we'll move now on to section 3. And for topic 3, which is called infection and response, the separate only topics are monoclonal antibodies, plant defences, deficiencies in plants, and for higher tier only, a little section about plant disease. The next section is on monoclonal antibodies. Let's start off by the idea then that we have something in our body, perhaps a bacterial cell or perhaps a cancerous cell that we want to target and get rid of. On the outside of cells such as bacterial cells and cancer cells, there will be antigens of a very particular shape. And you should know from this topic already that the white blood cells produce antibodies which are specifically designed to match the antigen. And what these antibodies do, these are produced by lymphocytes, which are special white blood cells, and these communicate and signal to the other white blood cells, which are the phagocytes, to come and engulf the bacterial cell or the cancer cell or whatever it is we're talking about. So here we've got the antigen of a very specific shape. This is the antibody, which is produced by your lymphocytes. And when this signals, when it's latched onto the bacterial cell or the cancer cell, it will signal to the phagocytes, and they will come and do phagocytosis, and they will engulf the pathogen and get rid of it. So scientists know a lot about how the immune system works, and they thought, wouldn't it be great if we could produce loads and loads of these antibodies so that we can target these specific cells and destroy them a lot quicker. So that's where monoclonal antibodies come in. First of all, we have our little mouse here who helps us with this process. The mouse is injected with a specific antigen. So this is the specific shape on the outside of our bacterial or cancer cell. So for example, if we want to design a monoclonal antibody to treat cancer, then the mouse will be injected with the antigen for the cancerous cells. And when this mouse is injected with the antigen, it will start to produce B lymphocytes, which are special white blood cells which are going to produce the antibodies to match the antigen. So for example, here we can draw our B lymphocyte with our particular antibodies that it has produced, and these will match that antigen. Now, all the scientists wanted to do was harvest and take these antibodies off of here and collect them all up into a bottle so that it can be used to treat a patient that actually had the cancer. But the trouble was these B lymphocytes took ages to replicate. They're just not quick enough. So what they had to do was they had to bind it with a tumour cell, because tumour cells divide really, really quickly. So we've got our tumour cell, and both of these were used to make a hybrid cell that they called a hybridoma. So we've got the lymphocytes produced by the mouse, we've got some tumour cells, and those two are brought together to produce something called a hybridoma. Now this hybridoma has the best of both worlds because it divides rapidly because it's like the tumour cell, and it also produces our very specific antibody that we wanted to get from this whole process. So these hybridomas replicate rapidly and divide and make loads more hybridomas. So we'd have loads of these hybridoma cells all producing the monoclonal antibody that we want to harvest. And we'd have loads of them, and then finally we can purify this so we just have our monoclonal antibody that we want, and we can bottle that up and we can use that to treat a patient. So the clue's in the name really. Mono meaning all of the same, clonal meaning exact copy of our antibody. So we've got our mouse to produce our antibody first of all. That's bound with a tumour cell to produce the hybridoma, and then they replicate rapidly, and then we can bottle up and use our monoclonal antibodies. So scientists were really excited about developing loads of these monoclonal antibodies really quickly to treat a variety of different diseases. So what they really liked about it was that it targets specific cells. So it's not going to go and damage healthy cells in your body. You've created a very specific antibody, so it's only going to target specific cells. However, the trouble with it is it produces more side effects than they first thought, so sickness, headaches and things like that. So they thought because it was targeting specific cells that the patient wouldn't get many side effects, but unfortunately that's a bit of a downside. The scientists did go further though. As well as producing these antibodies, they thought of some really clever ways that they could use them. So for example, they bound different things to the antibodies. For example, drugs that kill cancer, or perhaps a fluorescent dye or fluorescent protein. So the reason that they did this is because the antibody here will specifically go and target the cancer cell, and it will take the cancer killing drug with it. So if we have it over here binding to the antigen, it will also take with it the cancer killing drug, which will then kill and destroy the cancer cell. Some cells in the body, the scientists don't want to destroy, but they just want to show up just to see where they are in the body. So that's where the fluorescent protein comes in, whereby the antibody would latch onto the antigen and then would glow brightly to show the scientists where the antibody went in the body and to show them where the specific cells were. So these monoclonal antibodies can be produced for lots of different things. For example, they can be used in blood tests, and the antibodies will be latching onto specific proteins in the blood and signalling to the doctors whether they are present or not. And another specific use is in pregnancy tests. So women can find out very quickly if they're pregnant by using one of these pregnancy tests, and all of them are a little bit different, but they work in a very similar way, and they use monoclonal antibodies. So the pregnancy test uses specific monoclonal antibodies to look for a particular protein that is present in the urine of women that are pregnant. So what they would need to do is on this end of the stick, they would put their urine sample, so they'd wee on this end of the stick, and on this end you have monoclonal antibodies, but they are actually attached to blue coloured beads. Now, as soon as the woman urinates on the end of the stick, these then become mobile and will move up the stick. And this will happen whether she's pregnant or not. Just the fact that of the urine going onto the stick, these blue beads with the monoclonal antibodies will move up the stick. And if she's not pregnant, these will just move through and nothing will appear here. However, if she is pregnant, there will be a blue line appearing on this end of the stick, just like shown here in the photograph over here. The reason this happens is because the monoclonal antibodies stick to the particular protein that is found in the urine of pregnant women. So if you're not pregnant, you will not have this protein, which is called HCG, but if you are pregnant, this will be present in your urine. And what will happen when you urinate on this end of the stick, the HCG protein and its specific antigen will bind to the monoclonal antibodies that are stuck to these blue beads. So you will perhaps have one there and one there, for example. So when these blue beads then move along, the reason why you get a blue line showing if you're pregnant is because these blue beads will then get stuck in this area here. Because these monoclonal antibodies that are at the end are stuck in place and they can't move. So when these blue beads move across, the antigens of the HCG protein will stick to the antibodies that are here, as well, of course, as to the blue beads that they're already stuck to. So it will look something a little bit like this. We've got our blue bead that's moved from here with the antibodies on it, and it's already been stuck to the HCG protein, and that gets moved along and then it gets stuck up here at this end, and you will have monoclonal antibodies here stuck to the HCG protein and then stuck to the blue beads. So the blue beads remain in this area, and that's why you get a blue line. If you're not pregnant, that means you won't have this HCG protein and the blue beads will just keep on moving and will end up over here right at the end of the stick. But if you are pregnant, because of these antigens here on the protein, they will end up sticking in this area, showing that you are pregnant. And it's pretty effective. This question is about how plants defend themselves against disease. One can imagine a 6 marker on this, because there's lots of things that you can write about using some key words. So there's 3 different types of defences. There are physical defences. They are things such as the waxy cuticle, which is at the top of the leaves, if you remember the cross section of the leaves. And in defence, they are acting as a barrier to pathogens, so stopping pathogens getting in and infesting the plant. If they do get through the waxy cuticle, the plants also have a cell wall that is made from cellulose. That is to strengthen the cell, but also it'll help again to act as a barrier for pathogens from actually getting into the cells. Many plants also have dead cells around them to protect them from insects and smaller microorganisms like pathogens attacking them. So many trees, for example, have bark to protect them against a variety of different animals trying to eat them. As well as physical defences, plants also have chemical defences. So again, they can be attacked by bacteria. So some plants can secrete antibacterial chemicals to defend them against a bacterial infection. And some plants also contain poisons or toxins which deter animals and herbivores from eating them. So we looked at physical and chemical. The final type of defence is mechanical, and those are things such as thorns or hairs on plants to prevent them being eaten by all sorts of different animals. Other plants respond to touch, and they have leaves that can curl or droop if they are touched, and they're pretty cool plants. If you see this one here, when this gets touched by a human or another animal, the leaves actually curl up in a defence mechanism to prevent them from being eaten. The final defence then, which comes under mechanical, is mimicry. For example, these things here that look like pebbles are actually plants, but they're mimicking stones or pebbles to prevent themselves being eaten. This plant over here has yellow dots on the leaves. Again, this is a form of mimicry because to butterflies it looks like another butterfly has already laid its eggs here, and therefore that will prevent other butterflies from laying eggs. And of course, the first stage of a butterfly is going to be the caterpillar, which will come and eat all of the leaves up. So by mimicking butterfly eggs here, the plant is defending itself from butterflies and from caterpillars eating up the plant. There's two types of iron deficiencies that you need to know about in plants. The first one is a nitrate deficiency, and if you link your understanding here to the topic on how plants use glucose, you'll remember that nitrates are used along with glucose to build amino acids, and amino acids are the building blocks to make protein. And the final link, protein, is for growth and repair. And in this case, if the plant is deficient in nitrates, which means it doesn't have enough nitrates, because it can't build enough amino acids and proteins, you therefore have stunted growth. So this plant here, you can see compared to the other one, it has stunted growth, and that is an indication that it has a nitrate deficiency. The plant over here has a magnesium deficiency, and instead of stunted growth, you can see that the outcome of this is having yellow leaves. This is because magnesium is needed to make chlorophyll, which is the green pigment in plants. It's the one that absorbs light for photosynthesis. So magnesium is needed to make chlorophyll. So if the plant is deficient in magnesium, they won't be producing enough chlorophyll to make the leaves go green, and instead they will go yellow, and obviously that will reduce the amount of photosynthesis that the plant can carry out as well. So the last section is for higher tier only, and it's on plant disease. So if you are doing foundation separate science, you can stop there, but I would recommend you taking on board the separate only content, because there's only a little bit of it and it's likely that it will come up in your exam. So a little bit more for higher tier only. It's not a particularly difficult topic, plant disease, and I would expect you to be able to answer a 6 mark when it comes to the exam. So there's different symptoms of plants when they are suffering from a disease. So some disease, like the rose black spot fungus, for example, you will see spots on the leaf. That shows you that plant is suffering a disease. There may be areas of decay or rot on the leaf. You might see stunted growth, like we looked at with the nitrate deficiency before. There may be an obvious presence of pests on the plants, things like aphids and other small insects eating and destroying some of the leaves. There may be malformed stems or leaves, so leaves like this that don't quite grow in the correct shape for that particular plant. There could be growths on the plant, so sometimes you'll see really big trees in the forest with growths, large growths coming out of the tree trunks. That's a sign of disease as well. Discoloration of leaves, so it might not be magnesium deficiency, it might be another type of disease causing discoloration. And as a gardener, we would want to be able to identify what it is that's causing these diseases so that we can try and treat them or prevent them in other areas of the garden. It's especially going to be important for farmers and horticulturalists that are producing crops to sell on, because they're going to want the best crops that are free from disease. So once you've had a look at the symptoms of the plant, you might want to identify the plant disease so that you can treat it. There are 3 ways that you could do that. You could use a gardening manual or a website, have a look through the symptoms and try and work out what disease the plant might be suffering with. You could take it to scientists in a laboratory, plant biologists, and ask them to help you identify the disease. Or linking in with monoclonal antibodies, there are testing kits available that have specific antibodies which will help you positively identify the disease that the plant is suffering with. There are 3 main diseases and pests that you need to know about that affect plants. So we've got the tobacco mosaic virus, remembering that causes a mosaic pattern of discoloration on the leaves, reducing the plant's ability to do photosynthesis. We've got rose black spot fungus, a fungal infection, the symptoms of which are black spots all over the leaves, and they cause the leaves to turn yellow and drop off. And again, this reduces the amount of photosynthesis that the plant can carry out. And finally, you need to be aware of aphids, which are little insects that cause a lot of damage to crops and other plants in the garden. Aphids are really small, quite transparent little insects. They're quite difficult to see unless you're looking close up, but they can cause yellow spots on leaves. They can cause the leaves to curl and fall off, and they can do a lot of damage in a very short space of time. So a lot of the symptoms that we looked on the last page, leaves falling off or drooping, malformation of leaves, so leaves not forming properly, all of these things could be signs of aphids attacking the plants, and in this case you would have to buy a chemical spray or something to treat them, or if you're really lucky you'll have some ladybirds in your garden because they will come and eat the aphids and make the plants better for you. So there are no separate only topics for bioenergetics topic for paper 1, so all that we've covered before are the only separate biology topics for paper 1. So if you are doing a separate science, I do recommend you knowing these topics particularly well, because there's very few of them, but it is very likely that they will question you on some of these topics in your separate science exam.